Hello and greetings to everyone attending this webinar on antibody generation, challenges and solutions. My name is Glenn Marsowitz, and to briefly introduce myself, I received my PhD from Drexel University in Philadelphia in 2014. In collaboration with Thomas Jefferson University, we developed an antibody drug conjugate, specifically an immunotoxin against metastatic colorectal cancer. Since joining GenScript, I've served as the Antibody Services Marketing Specialist, working to bring our services, many of which I wish I had known about during my research, to customers like yourselves, making research easier. If you're not familiar with GenScript, we've been providing contract research since 2002. Beyond antibody development, we support all aspects of research, including catalog products, gene services, peptide and protein synthesis, and discovery biology services. As you can see, we also provide cutting-edge services such as CRISPR and constantly update our suite of offerings to stay at the forefront of research technologies. The result of this support is our status as the most cited contract research organization with over 10,000 citations and growing. Today's talk will be focusing on antibody generation challenges and solutions, first by discussing some applications for antibodies, then reviewing a typical antibody generation process, after which we will look into 3 possible challenges you might encounter during antibody development and provide a solution for each. And finally, I will outline some of our services which integrate nicely with these antibody generation solutions. So, just to briefly give an overview of antibody applications, which we will break into research and industry fields. Traditional research utilizes the directed specificity of antibodies to interrogate biomarkers within biological systems. Utilizing the standard assay of ELISA, FACS, and Western blot, researchers are able to tease apart target expression and function in tissue, even down to a subcellular level. Labeling of antibodies with a fluorophore allows for visualization of biomarker dynamics in real time with in vitro cultures, creating a truly powerful approach to understanding targetability, behavior, and ultimate fate of a biomarker. Antibodies can also be used not only as a means of measurement, but also as a manipulation tool. Antibodies may block ligands, activate or downregulate receptors, stabilize complexes, or mark cells for immune clearance through antibody-dependent cell-mediated cytotoxicity, or phagocytosis. In lieu of a genetic knockout or knocking an animal, these antibody functions may be used to turn off or turn on signaling pathways to reveal their role within a complex system. Industry use of antibodies follows that of traditional research, using them as evaluation tools to gauge response while developing pharmaceuticals. Recently, antibodies have emerged as a class of drugs unto themselves, exploiting the same toggle on or toggle off characteristics seen in the last point of classical research. Antibodies may be used as therapeutics even without modification. Additionally, they may be used as the targeting moiety in a conjugate format, delivering a cytotoxic payload for cell killing, or detection label such as an isotope or near-infrared fluorophore for visual detection. Further still, antibody engineering now allows for many formats such as single-domain antibodies and FADs, or fragment antibodies, which have the same binding capacity as whole IgG, but in a much smaller package. So despite having been around for some time, antibody applications and use continues to grow and mature. While there are some alternative methods of generating antibodies, most antibody projects still begin within the framework of traditional antibody generation. In order to understand the antibody generation and its potential difficulties, we first need to look at the mechanism of immune response. At its core, the process begins with receptors on B cells specifically binding to foreign antigen. The antigen is then internalized, broken into peptides, and presented on the cell surface via class II major histocompatibility complex, or MHC. This is done so that cognate T helper cells may recognize and bind the peptide via T cell receptors and release stimulatory cytokines, or stimulatory signaling through cytokines. Once activated, B cells begin expansion and differentiation into, amongst other B cell types, plasma cells. These plasma cells are then responsible for secreting the large amounts of antibody necessary for appropriate immune surveillance. The goal then is to generate this immune response with an appropriate immunization protocol. The initial immunization, or prime, begins the process as the first exposure of the animal to target antigen. Initial immune response may be measured via an ELISA using serum samples against plated antigen and compared to non-immunized serum samples from the same animal. High titer serums, or serum which contains a significant amount of immunoglobulin and therefore require multiple serial dilutions to see a decrease in ELISA signal, is the goal of any immunization. While the prime can create an initial increase in measurable immunoglobulin, subsequent exposures termed boosts are required to create a more durable high titer response. Repetitive boosts of antigen exposure should be performed until there is no increase in titer from the previous boost. At this point, it is time to move on to recovering antibodies. Polyclonal antibodies are the simplest to produce, as merely recovering serum from the host provides material usable for assays. Serum can be further purified with protein A or G to recover only the immunoglobulin population, or specifically purified against the immunized antigen, recovering only antigen-specific IgG. As polyclonal antibodies are the product of many various B cells and recognize multiple epitopes per antigen, they can benefit assays which require increased sensitivity. However, the heterogeneous trait of polyclonal antibodies may suffer from high signal noise due to increased chances of cross-reactivity. Additionally, polyclonal antibodies are a consumable resource. The quality of immune response between animals can vary, and recoverable serum volumes are limited. So once the supply of polyclonal antibodies is consumed, it may be lost forever. Monoclonal antibodies, on the other hand, can be expanded in culture, frozen, and sequenced for in silico preservation or even modification. Beyond the benefit of long-term use, monoclonal antibodies are created from isolated B cells, resulting in a homogeneous population of immunoglobulin, which targets the exact same epitope. Whether used as a tool to block or activate signaling, or to therapeutically recruit immune response, monoclonal antibodies are pointed and consistent. Monoclonal antibodies, however, require a bit more work to generate before they can be put to use. Rather than collect serum, lymphocytes from either the periphery or spleen are collected and fused with myelomas via electrofusion. The only fusion product of a lymphocyte and melanoma are desired. Therefore, a selection via hypoxanthine, aminopterin, thymidine, or HAT medium is performed. HAT medium blocks de novo DNA synthesis, and as myeloma cells lack the ability to recover from this blockage due to a thymidine kinase mutation, non-fused myelomas die off. B cells have functional thymidine kinase but are non-immortal, so they too die off. Therefore, only the fusion product of a B cell with a myeloma possesses the required characteristics for survival, both a way to overcome aminopterin and immortal survival in culture. Once hybridomas are created, repetitive limiting dilutions ensure that single cells are isolated and allowed to clonally expand. At this point, supernatants from the limiting dilutions may be probed via ELISA for positivity. Selected clones should then be expanded and placed into factories for antibody production. Purification from hybridoma factories only requires immunoglobulin specificity, as target specificity has been accomplished through positive clone selection. With significant amounts of antibody recovered, it is at this point which each positive clone can be affinity tested, used in assays, or tested for functional effects. This, however, all assumes that immunization is no issue, positive clones result, and antibody performance is satisfactory. Often this is not the case. So let's look at the most popular responses received from the pre-webinar survey that can arise during the process and investigate their solutions. Here we see three typical issues that come to mind when thinking about antibody generation. First, creating a sufficient amount of appropriate antigen can be challenging. Second, generating a significant immune response to an antigen with high homology or self-tolerance could prove difficult. And finally, even if enough antigen is generated and appropriate immune response is created, the resulting antibodies could have low affinity for the target. Let's take a look at each of these issues independently. Starting with difficult antigens, antigen production in vitro is a topic for discussion all unto itself, but briefly, production in vitro can be challenged by low yields, low stability, low solubility, or suffer from complexity that is just beyond the capabilities of a given system. Antigens can also be toxic, making the immunization a difficult balancing act between generating an immune response and killing the host. The solution to this host of problems is overcome by changing how and where the protein is made. Rather than create a large amount of protein and inject a bolus into the host, recombinant technology offers a possibility to have the host generate the target itself. Using appropriate vectors for host expression, plasmids containing the target sequence are coated onto gold or tungsten nanoparticles and accelerated into the host via pressurized inert gas such as helium, otherwise known as a gene gun. In this manner, genetic material is directly forced into the host cells, impacting skin, subcutaneous, and muscle tissue. The transfected muscle and antigen-presenting cells ultimately create the target protein and drive the immunization prime. Subsequent boosting with cells expressing the same target is recommended in order to fully activate B cells. From that point, typical antibody generation may continue towards polyclonal or monoclonal ends. The key to this is the in vivo generation of the target, which negates any in vitro antigen generation issues. Here we see a case study using DNA immunization to create an antibody against glucagon-like peptide receptor 1, which poses a few challenges discussed previously. It contains 3 disulfide bonds and post-translational modification at the N-terminus. Folding and glycosylation, such as this, may prove challenging with an in vitro approach. However, with DNA immunization as a starting point, we were able to generate an antibody which allows significant detection of GLP-1 receptor expressing cells by FACS, high titration above background via GLP-1 receptor plated ELISA, robust detection of protein in a Western blot comparing lane 1, which is DNA immunized antibody, to compared to isotype control detection in lane 2. And finally, strong immunofluorescence staining of GLP-1 receptor expressing cells compared to non-expressing controls. So you can see that the resultant antibody works in all standard assay systems. This, however, assumes the antigen is non-homologous to the host, which brings us to our next antibody generation challenge: overcoming self-tolerance. Self-tolerance is the mechanism by which the immune system prevents attack of host tissue and cells. Being able to discern between self and non-self is critical for maintaining defense against foreign antigens, yet not mounting autoimmune responses. While this system is thankfully in place in regards to normal physiological function and overall health, it is a difficulty when trying to generate an antibody in a host who shares high sequence homology with the target of interest. Here we see a diagram of central tolerance, which prevents the movement of self-recognizing T and B cells out of the thymus or bone marrow via apoptosis or receptor editing, as well as peripheral tolerance, which creates anergic B cells when T helper cells fail to provide co-stimulatory function. The key to overcoming this self-tolerance, then, is to provide the signaling normally present in the context of foreign antigens. There are a few immunomodulatory means to achieve this, including adjuvanation with appropriate cytokines or presentation of antigens in the context of viral vectors. As our technology is proprietary, the most I can divulge is that GenScript's approach is to modify the target antigen in order to provide appropriate non-self signaling, allowing full B-cell activation. We see here that the non-immunomodulated antibody response from a cohort of mice is nearly undetectable as opposed to the cohort on the right, all of which received immunoplus antigen modification and so significant specific antibody titers. Finally, let's suppose that generating antigen in sufficient quantities is trivial and self-homology is low, yet the resultant antibody has subpar affinity measures. High serum titers may have indicated a robust immune response, but this simply could have been the polyclonality contribution, not the presence of a few high-affinity antibodies. What can be done now? Rather than simply repeat the process and hope to catch high-affinity antibodies with another immunization, there's a method of driving affinity, or at least the variety of possible antibodies. This method uses germinal center-associated nuclear protein transgenic mice, or GANP mice. GANP is responsible for the maturation of B cells in the germinal centers of lymphoid organs. As a result, GANP overexpressing mice undergo frequent somatic hypermutations in the variable region of the antibody heavy chain, leading to a larger than normal repertoire of variable regions and potentially higher affinity antibodies. Here we see the variety of antibodies afforded by GANP mouse immunization. While there are potential low affinity antibodies generated, the affinity of the clones recovered from GANP mice reach at least an order of magnitude greater than that produced through traditional immunization. If you are familiar with phage display affinity maturation, the concept is similar. However, this approach does not require a phage display platform or antibody sequencing to accomplish. Merely a foresight in using these transgenic animals is all that is required to change in your immunization regimen to potentiate recovery of higher affinity antibodies. So to conclude, antibody generation is complex. This is a double-edged sword as it offers opportunities for failure, but also optimization. Novel techniques such as DNA immunization, immunomodulation, and GANP mice offer elegant solutions to previously insurmountable problems. Immunization protocols and preparation must be tailored to fit target characteristics. And finally, collaborating with experienced hands will smooth the application of these new technologies if you aren't familiar with them. I'd like to point out this time that GenScript has a few services which integrate nicely with these approaches. We offer polyclonal antibodies delivered within 45 days, monoclonal antibodies that are guaranteed to work in your application, phosphospecific antibodies that will not react with more than 10% non-phosphoproteins, and even anti-idiotype antibodies, which are ideal for antibody drug development, pharmacodynamics. The beauty of all these services is that we can customize them to include any of the technologies discussed today. We offer DNA immunization, ImmunoPlus, our proprietary self-tolerance breaking approach, and transgenic GANP mice. At this time, I'd like to thank you all for attending, and if you have any questions, please type them into the questions field in the GoToMeeting webinar tool. As you enter your questions, I'd like to remind you that we'll be sending a follow-up email that will help tailor our future webinars to your needs, so please look forward to it. Respond if you'd like to learn more. We have one last webinar set for May 18th at 9 a.m. Eastern Standard Time, covering recombinant protein production in baculovirus and insect expression systems. So sign up for that if you're interested. Finally, all our webinars are archived and can be found at genscript.com/webinars, should you like to revisit this or other topics. Now let's see if we have some questions. Okay, so a technical question here. What vector is used for DNA immunization? So we traditionally use ptDNA 3.1. It's a mammalian expression vector, but if you have empirical evidence for another one, we can obviously customize that service to your needs. Okay, a question about GANP mice. Do we have all rights to antibodies created in GANP mice? So I assume the antibodies that you create with them, yes, if it's a non-commercial use product, you don't have to pay anything else to us or the Transgenic Incorporated. However, if you're going to develop a commercialized product for sale, you would need to negotiate a license agreement with Transgenic Inc. They are the intellectual property holders of GANP mice. So I see a question about how the ImmunoPlus exactly works. So the ImmunoPlus is difficult to discuss in exacting detail because it is proprietary technology. However, I am at liberty to say that we modify the antigen itself so that it provides appropriate signaling during immunization. So the issue with immunizing with self-antigens is that you don't get the replete immune response and all the cytokine signaling. So if you can imagine a means of providing that signaling that is inherent in the antigen itself. So it's sort of like adjuvanation, but carried by the antigen itself. So hopefully that answers your question. I apologize that we can't be more specific, but it's a antigen-driven, antigen modification-driven approach to breaking self-tolerance. Right. Okay. I see. This is interesting. Can these approaches be combined? Yes. There's no reason why these cannot be combined. However, you would be very unlikely to require all of these. There's no technological issue with combining the approaches. I might add that since expression achieved through DNA immunization is usually low to start with the prime, higher affinity antibodies have been recovered with that approach alone. So you may not need to include the GANP mice as well. But as I showed, GANP mice merely widen the variety of antibodies possible. So if you're over concerned with affinity, you might consider both methods, but yes, they're all combinable. So there's a question about will the structure of the protein be maintained, and so I'm assuming this is in respect to the DNA immunization. And yet the whole point of DNA immunization is to have native structure presented to the immune system. The difficulty with a complex antigen like a GPCR is that, or any larger protein that requires appropriate glycosylation or post-translational modification or folding, is that by the time it gets to the immune system, there is not a uniform presentation of native structure. So by having the host express that structure in a mammalian system, if it's a mammalian antigen, provides the most native exposure. All right, so I think at this time, if there's no more questions, I will wrap it up and thank you all again for attending. If you'd like to contact me with any further questions you come up in the future, or come up with in the future, please email me. My name again is Glenn. Email glenn.marsalowitz, I know it's a long name, but it's correctly spelled here, at genscript.com, or contact anybody at GenScript. We can definitely answer all your questions in respect to this and all our other services. Thank you again for attending.